Start. So, hello everyone. It's nice to see some of you there. And today I would like to know something more about optogenetics and vision. So, optogenetics is made of two parts. The word itself is made of two parts. So, opto and genetics. So opto means it relates to light and means that something is activated by illumination, whereas genetics refers to genetic engineering. We mean genetically encoded protein. And the name of the genetics was first proposed for depolarization of channel rhodopsin 2 protein. The polarization of cells via this protein, yes. And so the genetics part usually refers to adeno-associated virus vectors because we insert in them these channel rhodopsin or other opsin proteins into these vectors, and so the cells that are depolarized, for example, in this case, were transfected with these vectors. And the biggest application of optogenetics was actually in neuroscience. So as I read this quite recent book, when we excite nerve cells, we can map neural circuits. Then a bit about those channel rhodopsins. I will talk a lot about them. First, optogenetics, first opsins introduced into the optogenetics field were holorhodopsin and bacteriorhodopsin. They are a bit different from channel rhodopsin because they are much simpler proteins. This is a pump and this is a proton pump. This was both of them are from archaeas, whereas channel rhodopsin is from green algae. Now, how these opsins developed? So as you remember from your photosynthesis lectures at school, maybe, algae and other microorganisms that photosynthesize produce energy by photosynthesis, so we need to move towards the light. And so by expressing those light activated proteins, bacteria and algae could produce energy for themselves. So there are type 1 opsins that are microbial, have microbial origin and are ion channels or ion pumps, and there are type 2 opsins that are much more intricate proteins. They are GPCRs, not simple channels, not simple pumps, and they are of animal origin. So these entered optogenetics first, as I mentioned, and bacteriorhodopsin is pumping proton ions, holorhodopsin is pumping chloride ions inside cells, so hyperpolarizing cell. Whereas channel rhodopsins are non-selective cation channels, and as you see, this heterotrimeric G protein is GPCR, and sensory rhodopsin was found in algae, but as I read papers, not much is mentioned about them, so I think that they aren't used much in optogenetics. Now, this is the heading of the paper about optogenetic approaches to vision restoration. So I found a very nice diagram in that paper that introduces you to concepts primary, secondary, and tertiary neurons. So as you know, light enters the eye and goes through retinal ganglion cells, bipolar cells, horizontal cells, photoreceptors, and reaches those photoreceptors that are also called primary neurons. Then from photoreceptors, signal is transduced to secondary neurons called bipolar cells, and from secondary neurons to tertiary neurons that are called retinal ganglion cells. Now, there are various inherited retinal diseases. I will talk about them later. And as I mentioned, vectors code for photosensitive proteins, and these proteins are inserted into retinal cells with the purpose of either converting bipolar or retinal ganglion cells into photoreceptors, or more rarely to resensitize degenerated primary neurons. These two goals are very important, and they will be necessary to remember in other slides. Now, I would like to talk about two major causes of blindness. I will not talk about cataract that can also cause blindness, but I will talk about retinitis pigmentosa. So it is a genetic disease, and it starts with rod degeneration and ends with cone loss. So as you see in this picture, here is healthy retina with cones and rods there. Then when a bit of rods degenerate, we are left with only cones, and later cones degenerate too. So here you see no primary neurons, and here you see remodeling of bipolar cells. Now, retinitis pigmentosa is not associated with 1, 2, or 3 genes that could be easily targeted with gene therapy. It includes 3,000 mutations, and they could be different in every patient. And these patients have irreversible blindness usually in this stage, for example, and therefore these patients are really wanted in clinical trials that want to restore irreversible vision. So after cones die, the central area is affected, and this leads to loss of color and central vision. Now, another cause of blindness is age-related macular degeneration. So macular degeneration refers to this area of the eye, which is a bit broader than fovea. And as you know, in fovea, cones are mostly located, whereas rods are located throughout all the retina, all the periphery of the eye. Now, is this macular degeneration a genetic disease? Well, half of the patients have some associated genes with it, but mostly this disease is environmental, like depends on your nutrition, healthy lifestyle, and mostly depends on the age, because highly the population that is at high risk of developing this disease are patients older than 65 years old. And this disease always produces irreversible blindness, and it also includes progressive photoreceptor loss, especially, and this loss leads to retinal epithelium degeneration. And there are two kinds of age-related macular degeneration. It's either wet or dry. So wet that type of age-related macular degeneration leads to neurovascularization, as you see in this picture. So when I do internship, not many people, but many projects are related to inducing this stage of disease. So we take a laser and the laser breaks, the laser penetrates the eye, and the Brox membrane is being broken, and here new vessels start to develop. And why does that lead to reversible blindness? Because these vessels can bleed and thus lead to blindness. So you see here retina, above is macula, and here you see retinal pigment epithelium layer.
Can I have a quick question about the
Yes.
previous slide?
Okay.
Do you have any idea what's the potential contribution of genetics or environmental factors?
Yeah, there will be those 3 genes related. I will talk about in a few minutes. Okay.
Perfect.
Good question. So that Brox membrane thickens when in the beginning of this disease, and when drussens develop, and this leads to edema and bleeding, as I mentioned. So Brox membrane is a very, very, very important membrane in the eye. It is located between retina and choroid. You see here is retina with its photoreceptors. Here is that epithelium layer. Here is the green one, the Brox membrane, and here you see choroid. And you see how drussens make the membrane distorted. They're really, really bad things. So retinal pigment epithelial cell layer is very also important because it contains metabolic enzymes, vascular endothelial growth factor, and platelet derived epithelial growth factor. Sorry, it's my mistake. And this is antagonist of vascular endothelial growth factor. And then there is balance between the two metabolites and also other secreted cells. Then there is homeostasis in the eye, but in this disorder, this homeostasis is diminished. There are more vascular endothelial growth factor than its antagonist. Therefore, it leads to neovascularization, which is not bad in this, which is not good in this case. And metabolic enzymes secreted by this layer metabolize vitamin A, which is precursor of cisretinol. And after this molecule is oxidized, we have cisretinol. And if light excites that cisretinol, we have transretinol, which activates photoreceptors. Now about genetic component of this disease. So cholesterol-related genes are thought to play a role because cholesterol makes up membranes, including Brox membrane. Certain mutation in this gene causes Brox membrane to be less permeable, and therefore blood can carry less metabolites to the retina. Also, smoking causes decreased transcription of complement factor H, and this increases the risk of age-related macular degeneration. And as I said, drussens that distort the epithelium hugely leads to oxidative stress in the eye, and this leads to accumulation of non-coding RNA ALU, and then inflammatory protein transcription is increased, specifically NLRP3 protein, and this leads to the epithelium we saw to undergo apoptosis. Then after macula degenerates, the lose central vision and most color vision. So yeah, it's because from macula, the image is sent to the brain. It's really important. The macular should be undisturbed and relatively dry for the brain to have clear and detailed images. And macula is one of the things that distinguishes humans from other beings on Earth. And diabetic retinopathy often affects the macula and leads to age-related macular degeneration. Now, about the treatment of this disease, including optogenetics, is intravitreal injection of the virus with the vector of channel rhodopsin 2 that is then expressed on retinal ganglion cells after some time has passed after the injection. Then we take a LED lamp and stimulate transfected cells. Google can enhance the light transmission into the eye. And then we record spike trains by retinal ganglion cells. And when we increase light intensity, we get higher firing rate. And the firings are transmitted through optic nerve to the brain. Now, there are many strategies for reversing blindness, and optogenetics is considered to be the most advantageous one. So without optogenetics, there are options like making pluripotent cells that differentiate, and we have photoreceptor precursors that are inserted into retina, mice or rats. But this works only in healthy tissue, not in diseased tissue. And also, the limitation is that there are a limited number of transplanted, like there is the capacity of the retina to accept engineered stem cells. Now, more advanced strategies are retinal prosthetic devices. And these are like small electrodes that are inserted into the eye, and they or directly stimulate retinal ganglion cells and restore sight to a certain extent. And this is exactly how cochlear implants work. We work by electrodes stimulating cochlear cells. And since they are successful, since people who lost their hearing can hear now, I believe that we could make blind people see something again. And in stimulating eye with electrodes is quite inconvenient, but new implants are wireless, and I will show you very nice pictures in the end of the presentation. Now, disadvantages of these prosthetic devices are very complex surgeries. Imagine you have to put small electrode into your eye. And then also it's why we still have irreversible blindness, whereas we don't have irreversible hearing loss, because retinas require more than 600 independent pixels to enable facial recognition. Another strategy to reverse blindness is to block potassium channel. When we have delayed repolarization of cells, so increase inaction potential duration, and cells are more excitable. And this was done with retinal ganglion cells in mice, and it hasn't been done in humans, from what I know. Now, if it was done in humans or when it was done in mice, it was not really convenient because you need regular intravitreal injections into your eye, or regular insertion of ocucerts that are things that you insert into your eye, and drugs diffuse slowly into your eye constantly. And another strategy of reversing blindness is gene therapy, but as I already told you an example, there are many mutations, rare mutations, and we simply do not know which gene to target. And also, even if we identify some genes, by targeting them, by taking them out, we only prevent vision loss, but cannot restore them. The loss of vision. Now,
I have a question.
Okay.
About the previous one again. So, how regular are we talking with inter-EVT, basically?
Yeah, it's an interesting question. I actually do not know. Probably that photoswitchable potassium channel blocker is metabolized in our bodies, so it depends on blockers' half-life, I would say. So if we have different blockers, probably there would be a different time after each IVT, I would say.
But it is not feasible exactly due to the reason that we need to do a lot of EVTs for humans in comparison with mice, or sorry, maybe I missed the point why it was not that feasible in humans.
Oh, why it was not feasible?
Yeah.
I think it's because of being not. Imagine, like, if you had to do regular IVTs, you cannot. No, it's not a eye drop. You can, do eye drop by yourself. But if you will need that IVT, I know, even every week going to your doctor, I think it's a lot.
Yeah.
I think it would be even more frequently than every week. Like, when do you take drugs? Like every day, every 3 days? I'm not sure about that regular, but I think that it's not so convenient. And there are better ways. For example, optogenetics, when you do it only once, and it's fine. So
yeah, makes sense. Thank you.
Then photopharmacology was a very interesting concept for me when I found this in this paper. So yeah, it's about the same thing we just talked about. So it's about photostitchable potassium channel blockers. So one of the blocker is cyclic azobenzene, and trans form is more stable than cis form, and in darkness, even that cis form isomerizes to low energy trans form. So how would it look like? So I don't know. Oh, it was intravitreal injection. So when azobenzene is in your eye, we would take a LED lamp and would illuminate the eye, and then this azobenzene would because I think it would be UV light probably. So either of this form of azobenzene, I'm not sure which one, would block the potassium channel. And if it was on retinal ganglion cells, for example, these cells would, as we talked, would depolarize and start firing action potentials. Therefore, we would get a signal in the brain. Now, there are also non-naturally photosensitive retinal neurons that we make into photoreceptors after we express opsins on those retinal neurons. So we can make actually any neuron into artificial photoreceptor. And the very good thing is that we can do this at any stage of disease. Either all cells are degenerated or only start to degenerate. Now, optogenetics treatment of eye disorders has some disadvantages. For example, phototransduction cascade is not so efficient as in healthy retina. If you imagine all your cones and rods are degenerated, and you express those opsins on retinal ganglion cells, so the light goes directly to retinal ganglion cells, and there are not so much processing of the light at photoreceptor level, and therefore the signal that is transmitted to the brain cannot produce a very efficient view as good as with healthy eyes. Then, yeah, so it's exactly what I said there. Maybe so we could have some goggles that could, through which light passing could be processed a bit, like resembling photoreceptors and rods. Maybe, I don't know, it's physics, maybe it works. Yeah, and there is no convergence, so we would need a greater light to stimulate those opsins. Now, when we want to treat eye disorder with optogenetics, we have to decide what opsin to express. As we saw, there are bacterial rhodopsins, channel rhodopsins, etc. And what cells we want to target. Are these cones? Are these rods? Sometimes they are not degenerated, they are just dormant. They sit there, but we are non-functional. Maybe we want to reactivate them. Maybe we want to express opsins on amacrine cells, on horizontal cells. Which one to choose? So we employ a molecular engineering and change opsins, certain opsins, so that, for example, they have increased light sensitivity, or they become from non-selective cationic channels to specifically, for example, calcium channels. So if the opsin allows chloride ions to enter, as we saw in bacterial rhodopsin, the cell will hyperpolarize. If opsin is a cationic channel, then its activation will lead to cell depolarization. So we have to decide if we want to depolarize or hyperpolarize cells. Then it's better to choose red light sensitive opsin instead of blue sensitive one, because blue light can become phototoxic, and thus it will lead to higher light intensity than needed to stimulate opsins, and it will lead to serious health risks. So rhodopsin was activated on bipolar cells or on melanopsin cells. Melanopsin is not actually a photopigmented cell, but it is still responsible for pupil constriction and regulates a sleep-wake cycle. So it's still involved in vision. It's very interesting. Even, you know, it's not the main cell in the eye, it still plays a role. So because of blue light phototoxicity, redshifted variant of channel rhodopsin was made with molecular engineering. Also, there are some other opsins developed, for example, calcium selective channel rhodopsin, or chloride conducting channel that was made by replacing negatively charged residues in the channel pore. Those negatively charged residues were present in unspecific cation channel rhodopsin, and after making adding more positively charged residues into this channel rhodopsin, we had a better fitted channel for chlorides to chloride ions to enter the cells. And also a bit modified, this channel was converted into I channel rhodopsin. So as you see, there are so many different subtypes of channel rhodopsins, and we should decide which one is the best one. So now about still about the same opsins. So these are class 1 opsins and class 2 opsins, and microbial opsins are advantageous over vertebrate ones. This is the impression I have made from reading some papers. So it's because microbial opsins are faster, they are autonomous, and they can be activated directly, and no cellular machinery is needed for them to transduce a signal. However, there is immunogenicity due to them, since it's microbial protein. Any human or mice or rats usually detect those proteins as foreign ones. And also another disadvantage is that we have lower light sensitivity, and advantage of microbial opsins is that renewal is less frequent. So I understand it as desensitization, that GPCRs undergo desensitization after we are used for activated for some time. They desensitize, they are phosphorylated so that we cannot transduce any signal for some time. It's not the case for microbial opsins. So vertebrate opsins, many of them, their characteristics can be explained that by the fact that we are G protein coupled receptors. So the response after their activation is slower compared to microbial opsins. The interesting thing is about transretinal recycling. As I mentioned, that retinal epithelium layer is very important because it contains enzymes that convert that very stable trans isomer into cis isomer, and then light can shine on this isomer and make trans isomer. And this isomer is very important because after we have this, we have activation of GPCRs and all activation of photoreceptor cells and all the firing from the eye to the brain. Now, activation of vertebrate opsins is indirect, cellular machinery is needed, and there is no immunogenicity. It is a good thing. Light sensitivity is higher, and renewal is needed after each photoisomeration. So why wouldn't we bring together all good sides of microbial opsins and vertebrate opsins to make one good protein? This is how ionotropic glutamate receptor was made, and some other proteins. Now, challenge of opsin expression on cells is that the expression should be stable, and the protein should be trafficked correctly on the cells we want to express, because I have read sometimes we want express the opsin of retinal ganglion cells, but somehow also bipolar cells start to express the opsin, amacrine cells and horizontal cells, whereas we wanted only bipolar cells to become photoswitchable. So this was ionotropic glutamate receptor, and the story is exactly about this. So also regular injection of the ligand of this receptor is needed. Light sensitivity must be in blue UV range usually, and melanopsin absorbs blue light, and its sensitivity was conferred to that metabotropic glutamate receptor on bipolar cells, and then retinal ganglion cells could fire signals. So this actually worked model in mice. Now, yeah, this is on bipolar cells. Now, bipolar cells is considered to be the best target of opsins, not retinal ganglion cells, as I told you. So we are very important cells because we are interneurons in retina. We connect photoreceptors and retinal ganglion cells directly. And we receive inputs from a set of photoreceptor cells when glutamate is released, and this defines bipolar cells receptive field. Now, glutamate release is decreased upon exposure to light. So after light is off, we have darkness. We have a lot of glutamate released by photoreceptor cells, and off bipolar cell is activated then. And why it is called off bipolar cell? Because it is activated when the light is off. You can remember it like that. The interesting thing is that we have only one type of rod bipolar cell and many types of cone bipolar cells. And in optogenetics, why we usually target rod bipolar cells? Maybe you have an idea. Why don't we target cone bipolar cells? It's because rod bipolar cells are much more prevalent in human eye. Remember I said that rods are prevalent across all periphery of the eye, whereas cones are only present only in the fovea. So you see here bipolar cell connecting photoreceptor cell with ganglion cells. Yeah, and the thing is that optogenetics always, in most cases, I have never seen a paper when off bipolar cell would be targeted to express opsin. It's always on bipolar cells because it is sufficient to generate on or off responses that either making expressing hyperpolarizing opsin on bipolar cells, we inhibit them, and this produces off response, or we can express depolarizing opsin on bipolar cells, so activating these cells. So all bipolar cells are not so important. Shortly, how on bipolar cells work. So after a light is shown, less glutamate is released, as I already told you, and from cones. Then this leads to activation of inhibitory metabotropic glutamate receptor, and because not much ligand, the receptor is activated not so much too, and this leads to less inhibition on bipolar cell. So bipolar cell is more activated, and because this cell is not inhibited, so it fires more, it releases more glutamate, thus depolarizing bipolar cell ganglion cell very well, and there is more firing, and so we have sign-inverting synapse where we started with less glutamate, less excitatory transmitter, and we ended up with excitatory ganglion cells and a big signal to the brain. So as you see in this picture, this is how everything started in 2006. It's all about channel rhodopsins, mostly are channel rhodopsins. And retinal ganglion cells were targeted mostly. Later, as you see, in 2010, 2015, only on bipolar cells were targeted, mostly, not once retinal ganglion cells. But as I mentioned, on bipolar cells is much better because I think that promoter is more accessible or more known or more studied than yeah, it just works very well when we do not send conflicting signals into the retinal ganglion cells. And also, I also already talked about this one. And yeah, so the proof of this is that blocking on pathway, blocking on bipolar cells in rhesus monkey caused impaired contrast sensitivity and restored the pathway. And this was enough, but not in retinitis pigmentosa, the first disease I told you that is genetic disease, and more than 2,000 genes can cause this disease. So this is about a bit about retinal dystrophies. So in these diseases, cones have lost their membrane protrusions, for example, outer segment. And this outer segment is relevant for phototransduction. So in dystrophies, rods cannot phototransduce signals, and the cones are called dormant. They are not light sensitive. And this is the case in blind patients, exactly in that macula of the eye, as I showed you near the fovea. In 2010, these dormant cones were reactivated, and actually retinal ganglion cells fired the signal, and people could record the signal. Microbial opsins were inserted into human post-mortem retinas. So there was no immunogenicity somehow, probably because these were only retinas and not human alive. An advantage of reactivating dormant cones is that we do not lose photoreceptor processing. We do not need the googles that would compensate from that processing of information. And in this case, we need a bit deeper injections in the eye that looks like that, not intravitreal injections. So now why bipolar cells are better than retinal ganglion cells that were targeted first? Well, bipolar cells are preserved in degenerative retinal diseases. Very close to the photoreceptors as compared to retinal ganglion cells. And also they are larger in number compared to retinal ganglion cells. So this is similar why we target why we talk about more about rods than cones, because rods are more prevalent in the eye than cones. And also bipolar cells do not have lateral extending processes that provide focal activation. Now what's left? 5 publications to talk about. So I already mentioned the first one. The thing is that it talked about inherited retinal diseases and provided a very nice statistics. Yeah, and it also it's kind of review, so just to be up to date. Actually, electronic retinal implant is reality, and it was approved by FDA in 2013. very long ago. A gene replacement therapy was also actually approved in the USA when electronic retinal implants are limited still in their maximum achievable resolution and provided only limited field of vision. This is why we need optogenetics to restore human vision to a bigger extent. And again, the same problem, we don't know the genotypes of people that have a reversible vision loss. And yes, it's the same thing we talked. And we need to convert secondary or tertiary neurons into photoreceptors. That is usually what is usually done. Now, this paper is a very recent one, is 2020. And it was published in Nature. The reason why in Nature, I think, is because it presented a new protein we didn't see in the table. It's not channel rhodopsin, it's multi-characteristic opsin, and it is sensitive to AMB at night. So it feels you wouldn't need LED lamp to illuminate your eye. Since that opsin has lower threshold required for optogenetic stimulation. Then in that study with mice, absence of inflammatory response was observed. That is a very, very good point. And no change in the structural eye architecture were observed after injections, as we saw in retinitis pigmentosa. After rods and cones are degenerated, bipolar cells start to rearrange themselves, and that's bad sign, and there was no such bad sign. And on bipolar cells were transfected with that multi-characteristic opsin by the cell-specific promoter, that inhibitory glutamate receptor. Then the problem this research wanted to solve is again that retinal prostheses have been unsuccessful in making disappear irreversible blindness, and limitation of prostheses include cellular outgrowth, chronic damage of implanted electrodes, and insufficient or disordered stimulation of retina. Then a bit of outside the vision, just focusing on a recent papers in optogenetics. It's about enhancing memory. So it could be after traumatic events in diseases such as Alzheimer's or diencephalic amnesia. And also optogenetics could be used to diminish the cravings of addicts. And this paper was very, very interesting for me because it contained paragraphs like that are purely philosophical. For example, role of values were emphasized for authenticity. And the issue that was discussed in this paper was that if modifying people's memories using optogenetics is ethical due to influencing patients' values. Yeah, because optogenetics could disrupt personality of patients. Now, quickly, just how optogenetics looked in time. So Geramis and Bach activated neurons by light in 2002. So this optogenetics field is very, very recent, actually. And retina was reactivated in 2006 using optogenetics. In 2007, mouse could walk in circles after shining the light. when in 2011, silencing orexin hypocretin neurons caused slow wave sleep in mice, and pain excitation or inhibition could be caused by optogenetics in moving mouse in 2014. Now, as you see, Vilite was delivered transdermally, for example, in these mice, but also we will see later that we can put the electronic device into the brain that contains light, and after you press a button, the light is shown, so that you do not need external light application onto a human or a mice. So now a bit about the sleep. So it's fourth paper I would like to discuss here. So the very, very huge problem that optogenetics would solve with sleep, in sleep is that existing drugs now usually repress REM sleep and the deeper stages of non-REM sleep, REM sleep. And you know, REM sleep is very important for our memory. So people who suffer from insomnia may have impaired memory just because of the drugs being, I would say, ineffective in producing qualitative sleep. And then researchers generated transgenic mice where orexin neurons were made to express halorodopsin, and orange light was shown on those neurons, and the neurons hyperpolarize, and orexin neurons could not discharge actin potentials, and this led to the synchronization of the electroencephalogram and the reduction in amplitude of the electromyogram. And this meant that patients entered slow wave sleep. And it was not clear what is the function of orexin neurons. I think it's not clear yet. But after this optogenetics experiment, we can suggest that orexin neurons play a role in sleep, in REM sleep, but in deeper stages of sleep. And the last paper that I think is the most fascinating one is very recent, 2021, January 22. So a subdermal implant capable of wireless battery charging and programmable controls for applications in optogenetics. So it solved a very, very, very huge problem that before this day, optogenetics devices had to be tethered to participants for light delivery, and that restricted animals' movement and caused increased inflammation in the soft brain of the tissue where these electrical wires were. And their device is such that there is no need for power transfer. For example, these mice are in this cage and you just press a button and the battery is being charged, and so the LED, there is no need for LED lamp because LED lamp is also in mice brain. And this is what is inserted into mice brain. It's a small device that contains LED lamp and it has battery, it has Bluetooth system on chip, and wireless power management circuit. So it feels that it can activate or the thing was these mice were tested with ketamine, so we were given ketamine, and after that locomotion response usually occurs, but by activating this device, no locomotion was seen for 1 hour. So this method was proven to be really, really effective and is very promising strategy. It feels like if you can press a button and your, for example, bipolar cells can fire, you can feel contrast, so that blindness can be reversed, at least to some extent, and you don't have wires from your brain, because this is just implanted by a surgery into your brain. So thank you for your attention, and maybe you have some questions.
Can I ask you if you know how do they like exactly do the cognitive enhancement in the optogenetic study?
Sorry, we do what?
Cognitive enhancement.
Oh, cognitive enhancement.
Sorry.
Okay. So I think there are certain neurons. So simply by increasing the firing of certain neurons or decreasing the firing of certain neurons, we should know what neurons we want to induce to improve our memory. So if you know, then I think it could be done, but I am not sure. Yeah, it's quite recent field. It's 2019. We should look in that.
Cool, thanks.
And do they do all of the 4 things mentioned in just one study, or is it like an overview, or do they actually view all of the 4?
Yeah, I took this from introduction. So this was
okay.
Yeah, so they cited this paper, and there is, I think this paper, this paper I think is 2020. So yeah, this is quite new field.
I haven't heard of optogenetics being used in psychiatry before, and this is really exciting. Thank you for finding this paper.
Yeah, that's interesting. But it's quite controversial. I don't think that it could be implemented on humans. Like,
yeah, it is. Yeah.
How about oscillations, Nastya? What do you think if they can alter oscillations and then like gamma band stuff? So cognitive enhancement.
I'm not sure it's actually feasible with optogenetics because, you know, you kind of need to stimulate a lot of neurons at the same time. But I'm not sure. I think you need to have like a huge, I don't know even how to call this thing, which you like implant into the brain to shine on a huge area of neurons. And I'm not sure if it's like going to be more controlled rather than direct electromagnetic stimulation. I don't know, but yeah, it's really interesting. I had no idea.
It's also strange that in Alzheimer's disease, most neurons are degenerated. So it's interesting if we are dormant and we can reactivate those neurons. Maybe this is how the memory enhancement works. But I think that in Alzheimer's disease, I thought that neurons are degenerated and we are just absent. There is nothing to reactivate. I am not sure.
Yeah, exactly. And also diminishing the craving of addict. I think it might be something involved with dopaminergic system, but I'm not sure. I think it would probably involve actually shutting down the dopaminergic system. But
for example, in this case, this was carried out with ketamine. This was proven to be effective with ketamine in ketamine is an NMDA receptor antagonist, right?
Yeah.
So could be
Wow.
dopaminergic. But it feels that something is gonna happen. So maybe we will have no irreversible blindness. Any more questions, or we can end this meeting? So please send comments, please send feedback to Messenger, or if you want, you can save her, so that other times we could have better quality presentation.
Can I just thank you for this presentation? I think it's the best journal club I've ever been to. Like seriously, usually people just review like 1 or 2 studies, but this is just some quality lecture, you know. I think I will, yeah, I will definitely come back to the recording and like watch it all over again, because my dissertation is actually connected with optogenetics. So there's so much useful information for me here. So thank you so much. This is like next level impressive.
Take it with a pinch of salt. I'm not an expert. So, you know, I'm not an expert in this field. I just reviewed some papers. So thank you very much for your attention, for listening, and I hope to see you in other journal clubs. Bye, Ben.
Thank you, everyone.
Thank you very much.
Thank you.
Thank you.
Thank you.
Thank you.